Good evening, everyone. Welcome to Botany Insider's new marathon class that we are conducting on daily basis. So let's wait for the students to join in and then we'll start off with our class, right? We'll be discussing about the Drosophila, the next segment in the Drosophila topic. If you have not yet watched the first part, so you'll get a link of the playlist. So in that playlist, you'll be able to check the all the free classes that are available. Okay, so yesterday we had discussion regarding the what Drosophila is, what is the basic life cycle, and the related concepts. Good evening, Anjali. Let everyone join in, then we'll start. Just confirm whether the audio and the video is clear to everyone or not. Okay, so let's begin with our class now. Before starting the class, if you're new to Botany Insider, please make sure to hit the subscribe button. In addition, we have our own application. You can download our application in which, if you're structurally preparing, like if you wish to have a structured preparation, dedicated preparation for your December examination, we have a full course and we have a crash course running for the time being. You can enroll in the same for making your preparation more structured. In addition to that, you can order your notes booklet specifically curated for the December examination via WhatsApp either, or you can place your orders via application as well. Okay. So, and we're about to start with the PYQ course for the December 2023. It would be a revision plus a PYQ course. So those of you who wish to enroll in the same, you can download the application, and like we're about to start with the enrollments of the same within a day or two. So you can download the application before that, right? Let's begin with our class. So if you remember, yesterday we had a discussion that what Drosophila is all about, right? What is the basic framework of Drosophila? How does the life cycle of the Drosophila actually operates? Then we had a discussion related to the type of the life cycle. It is holometabolous in nature, right? That means it shows complete morphogenesis, complete metamorphogenesis in its body structure. Then we had a discussion that there are a number of genes that are involved for the developmental process of your Drosophila, right? What are the various developmental processes, or what is the entire complete group of the various genes that are involved? So you have the maternal genes, you have zygotic genes, you have homeotic selector genes. Remember, all these three categories work in a sequential manner. Initially, you have the maternal genes, then you have the zygotic genes working, and then you have the homeotic selector genes working in. The maternal genes are the ones which help in the initial, like the very first work, the initial pathway initiation is done with the help of maternal genes. Then you have the zygotic genes, then you have the homeotic selector genes. Okay, so in case of your zygotic genes, you have three subtypes. First is your gap genes, then you have pair rule genes, and then you have the segment polarity genes. Okay, so first maternal genes will activate, then after their functionality have got completed, zygotic genes will get activate. Zygotic genes will be like, in the zygotic genes you will be having gap genes, pair rule genes, and the segment polarity genes. And then after, like, these three will also be getting activated in a sequence. Okay, and after this you will be having the homeotic selector genes working in. Okay, so keep a note of this particular thing. Now when we look into the entire listing of the various genes that are there, so you have the maternal genes. In the maternal genes you have bicoid, hunchback, nanos, and caudal, which are the three genes, four genes, bicoid, then you have hunchback, then you have nanos, and then you have the caudal. Okay. Then you have the gap genes. In the gap genes, there are five main gap genes. You have to remember each and every name. Okay, you can make any mnemonics, whatever way you want to learn, but you'll have to keep a note of all the genes that are involved in the entire developmental process. That is, you have cripples, you have crims, then you have giant, hunchback, and tailless. Then in the pair rule genes, you have hairy, even skip, runt, fushitarasu. Then you have odd skip, slopey, paired, and paired. Okay, you have to remember the entire naming, all the genes that are involved in each and every group. Reason being, in the examination, sometimes you directly get a question in match the following type, where on one hand you are written down, you are given a number of genes, and on the other hand you are given their main groups that are involved in. Okay, so keep a note of it. Maternal genes, then you have gap genes, pair rule genes, then after that you have the segment polarity genes. In case of your segment polarity genes, we have wingless, enrailed, patched, hedgehog, armadillo, and cubitus interruptus. You have to remember all the names. Okay, first one is wingless, then you have engrailed, patched, hedgehog, armadillo, and then you have arbutus interruptus. Then in the case of your homeotic selector genes or the homeotic genes, in this you have 8 of the genes involved. First one is labile, antennipeda, sexcom reduced, deformed, proboscipedia, ultrabithorax, abdominal A and abdominal B. I cannot insist upon this particular terminologies, that the entire listing, this entire list of genes is very, very important. Not just in CSIR, if you're giving any other examination where you get deaf bio as a topic of or as a syllabus. So this particular topic becomes, or this particular topic is very, very important for that particular thing. Okay. So make sure that you are learning, that you are keeping a note of all these things. Okay, so repeating them again, the first one is maternal genes. In the maternal genes you have bicoid, hunchback, nanos, and caudal. These four genes are the ones which plays important role in the first condition, the anterior posterior axis formation. Second condition, ventral and dorsal formation, right? Ventral and dorsal axis formation. Then you have gap genes. So in the gap genes, you have cripple, crips, giant, hunchback, tailless. Then you have pair rule genes, all these names, segment polarity, all these names, and homeotic genes, all these names. Okay, you have to keep a note of all of these factors, right? Quickly go through this, then we'll proceed ahead. If you find any difficulty, you can ask. Okay, thank you so much, Kalpana. Thank you so much for wishing. Everyone, go through this. Very, very important. All these genes name are important. What you can do, you can just write down the entire listing of the genes, and then you can paste it in front of your wall, wherever you're on the front of your study table. Okay, so it will help you remember, or it will help you keep a note of all these factors, all these things in detail. Okay, quickly go through this, then we'll proceed further, everyone. Done. Yes, understood till here. Should we move ahead? Right, these are all the genes that you have to keep in mind in terms of your Drosophila segmentation gene, right? Then you have like what are the various sequential events that we have. So we have a number of sequential events that are there for the entire formation, for the entire understanding of your development of Drosophila. First thing is the fertilization. Then you have the cleavage. Then you have gastrulation, axis formation. In the axis formation, there is anterior posterior development, and then there is dorsal ventral development. Okay. And then you have the body segmentation, right? In the body segmentation, you will see the gap genes, the pair rule genes, and all the other genes working in case of your anterior posterior and your dorsal ventral, you have the maternal genes working in. Okay, so this makes the genes also very very important. Like again, this means that this complete segment is very important, because in the examination you have got question from fertilization, cleavage, gastrulation, axis formation, body segmentation. Like from each and every segment, from every single segment, questions have been asked previously. So this makes this particular topic very very important. Okay, so talking about the fertilization, so in the case of fertilization, one thing that you have to, or the most important thing that you have to understand is that obviously you'll be having a male partner, you'll be having the male donor, and then you have the female, right? The male will be uh, producing the sperms, and in the egg it will be going and fertilizing it. So let's say this is the female egg, okay, and the area or the point at which the male sperm enters is given the name of micropyle. Okay, what is it given the name? It is given the name of micropyle. So when the sperm enters into the egg via the help of this micropyle, there is asymmetrical or uneven distribution of your various uh, maternal genes. Initially, you have the hunchback, caudal, nanos, and bicoid, all of them getting segmented. Like equally, there is no proper set pattern for the same. But as and when from the micropyle portion, the sperm enters into this particular egg and fertilizes it, the hunchback increases, or like there is increase in the concentration of the bicoid onto the anterior side, and onto the posterior side there is higher concentration of caudal and nanos. Okay, in the side where you have higher concentration of, where because of the asymmetrical distribution as a result of this fertilization, there is increase in the concentration of the maternal gene or the maternal factor, bicoid and hunchback onto the anterior side, and onto the posterior side there occurs higher concentration, or there is high increase in the concentration of your caudal and your nanos. Okay, and because of this asymmetrical distribution, there is anterior and posterior segment formation, and this is the result, and this leads to the axis formation in the Drosophila, because of the because from the point of entry the sperm is entering in, that is a micropyle. From there, as and when the sperm enters, now not the sperm, it is the genetic content, it is the nuclear content which will be entering and fusing with the female part, right? And then there is initially uneven distribution, but as and when the male content enters in, there is a very segmented division of the various maternal genes that we have, because the genes will be causing the initiation of the transcription factors, right? Because the transcription factors will start to synthesize in the cell afterwards. So there will be higher concentration of bicoid and hunchback onto the anterior side, and onto the posterior side you have the caudal and the nanos. You can understand it the other way round as well. That is, the anterior side gets formed, or the anterior axis gets formed in the region where there is higher concentration of hunchback and bicoid, and there is A posterior side gets formed in the area where there is higher concentration of caudal and there is higher concentration of nanos. It's a very important concept, okay? And almost every second year you can easily find a question from this particular portion. Take a minute, understand this, then we'll be understanding the further portions, okay? Understood till here, everyone? Any doubt, any problem? Understand this concept properly, okay? And if you want to learn in detail, if you want to learn in more proper depth regarding all of these things, we have a complete crash course running. And at present, we are discussing this unit number 5 itself. So you can enroll in that and you can get to learn a lot and in much more greater depth, okay? If it's clear, then let's move forward. Yes, 14 of you are there in the class right now. Participate. Right, let me show you one diagram. It will make this particular topic more clear. So see over here, the sperm entry occurs at the micropyle end. Egg has to be like there's a specification of the axis, like as and when or the at the point at which the uh, entry of the sperm occurs, you have the specification of the axis is occurring. Okay. Now the stored mRNA starts the translation. Check over here. See, before the fertilization, what do we have? Before fertilization, you can see the hunchback and the caudal being in the normalized, like high concentration throughout. As and when the fertilization occurs, what do we have? As and when the fertilization occurs, you'll see a higher concentration of the hunchback in the anterior side and the concentration in the posterior side decreases. On to the other hand, you have the caudal concentration higher in the posterior side and it decreases on the anterior side. So this is the main concept behind the anterior posterior axis formation. Very, very important. Okay. This is something that gets repeated in the examination, and this is something which many of the students fail to have a proper understanding of. Okay, so try to clear out your concept now itself. If you find any difficulty, you can ask. Okay, use this particular platform to get your doubts cleared. If you're finding any difficulty, you can ask. It's a very, very important concept. Yes, understood till here? Before the fertilization, you can see the hunchback and the caudal being in the higher concentration. But after the fertilization, you can see the hunchback being higher on the anterior side, and onto the posterior side, you can see a dip in the concentration of the hunchback. And the other way around for the caudal also. Like for the caudal, there's increase in the concentration in the posterior side and there is a decrease which occurs in the anterior side. Okay, and as a result of the same, as a result of higher concentration of hunchback and bicoid in the anterior, there is anterior axis formation, and the other way around is the posterior axis formation, right? Clear, everyone? You people are not interested in studying today, right? Don't want to study today? It's such an important concept, but you're not participating in the class. Okay, so I hope till here it's clear. Tomorrow, we won't be continuing with this developmental biology because you people are not participating. So we'll be having some other topics discussed in the live sessions. Okay, so this is about for this particular class. I hope it's clear. You can download our application for much more understanding or a much deeper understanding for the entire concept. We'll meet in the next class with a new topic where we'll be discussing about the practice questions or the other topics. Okay. Bye everyone. Take care.